Hi, my name is Lei-Lei Kim, and I'm presenting on Legionnaires disease. So, how was the disease first discovered? In July of 1976, 4,000 members of the American Legion met at the Bellevue Stratford Hotel in Philadelphia to celebrate the 50th annual Legion Convention with their families and friends. So, this was a pretty big gathering. And then on the next day after the convention, a few Legionnaires started showing symptoms similar to pneumonia, like high fever and coughing. By early August, news organizations fled to this scene where they reported on this new mysterious disease, and they were calling it Legionnaires' disease after the convention. When the epidemic was finally over by mid-August, it was determined that 221 individuals contracted the disease and 34 of them had died. So then the issue was, how did this disease started? And CDC scientist named Joseph McDade and his colleagues identified the bacterium that caused the disease by January 1977. They isolated the bacteria by a technique using guinea pig inoculation. They named this infectious bacterium as Legionella pneumophila, and they discovered that the bacteria spread through the air conditioning system at the hotel, and this is the Bellevue Stratford Hotel. And after the outbreak of disease, and they had to close because of bad publicity. So the bacteria that causes Legionnaires' disease, as I earlier mentioned, is known as Legionella, but most notably caused by Legionella pneumophila. So this species actually caused about 90% of the reported cases, but different species in the genus Legionella can also cause the disease. And so Legion pneumophila has actually 15 different serogroups, and serogroup 1 is responsible for roughly about 80% of reported cases. Legionella are known as gram-negative. They are considered aerobic, and that's important later on, I'll mention. And they're non-endospore forming bacilli, and they have monopolar flagellum, which helps for motility. So pathogenesis. Legionella bacteria are intracellular organisms, so they infect the cells. And so when they enter through your mouth and then go to your lungs, they would attack alveolar macrophages, which are human immune cells in the lower respiratory tract of the lungs. And these bacteria would bind to the macrophage by complement receptors, and then they would be engulfed into a phagosome. And the way that they are not detected is by preventing phagolysosome fusion, and they do this by actually hijacking a protein called RAB1. So the enzymes in Legionella bacteria would alter the function of RAB1 and make RAB1 proteins remain on the surface of the phagosome. And once they do that, the RAB1 would attract transport vesicles in the cell, and these vesicles would cover the whole entire phagosome, hiding it from the lysosomes in the macrophage. So in other words, they would hide, the phagosome is hidden from the cell's defense systems, which is really cool, because then the bacteria can continue multiplying and growing, and then it would break open and repeat the cycle to other cells. And so because I said that it happens in alveolar macrophages, this disease is considered a respiratory disease, which makes sense because the bacteria is aerobic, so it would happen under oxygen conditions. So the immune response is kind of unclear, but they focus upon cell-mediated immunity because Legionella is an intracellular pathogen. So what happens is that lymphocytes become stimulated, and they would start to produce cytokines, which are known as lymphokines. And these lymphokines then start to trigger T helper cells. And so it would trigger Th1 helper type cell, and that would secrete some more cytokines known as interferon gamma and interleukin 2. And these lymphokines would then activate the macrophages to phagocytize and kill the bacteria. And then also humoral immunity plays a small role, but it's still kind of unclear about that too. But Th2 helper cells start secreting interleukin 4, 5, and 6. And these cytokines would activate B lymphocytes, and they would produce humoral antibodies, and that would and one of the functions of antibodies, as we learned in class, is opsonization. So they would start to function in opsonization, which will help activate the macrophages to phagocytize. So transmission, Legionella is actually found ubiquitously in fresh water all over the world, but the growth is not high enough to infect anyone. So they actually grow more in water systems that are man-made environments under like certain conditions, like they have the right temperature range in the water systems, or the absence of disinfectants. They can contract. People can contract Legionnaires disease when breathing in small droplets of water containing Legionella in the air. And so people then do not spread this infectious disease to other people. And other forms of transmission is aspiration. And this is really interesting because like a person can be drinking water that contains Legionella and they will not contract disease. But if they like drink the water that has Legionella and start coughing or choking, and then it gets into the airways, then they can have the disease. And soil is also known to transmit the disease. There were a few cases that people were like gardening, and then they were using potting soil, and they would start inhaling the soil that's mixed with water and start inhaling the dust particles, and the dust particles would have Legionella, and then they would contract the disease. So some common sources of transmission, as I mentioned, is usually man-made water systems. So that includes shower heads, sink faucets, hot tubs, decorative fountains, water features, hot water tanks, heaters, large and complex plumbing systems, and cooling towers and air conditioning systems, which I mentioned earlier in the American Legion Convention. So some symptoms include, so the incubation period for symptoms is typically from 2 to 10 days after exposure. The initial symptoms include headache, muscle pain, weakness and fatigue, loss of appetite, and then more symptoms will start to develop about 1 to 2 days after the initial. So that includes coughing, shortness of breath, chest pain, diarrhea, nausea, and vomiting, and confusion. And if this disease is not treated quickly enough, then it can become fatal, and that can lead up to respiratory failure, septic shock, which reduces the blood flow to vital organs like brain and kidney, and acute kidney failure. So the diagnostic tests include urinary antigen tests, which is actually the most common, but it can only test for Legionella pneumonia serogroup 1, but there's other types of species and serogroups that can also cause the disease, so it's not that reliable. And then there's also culture from the lower respiratory system that you can test for. And then the best test is actually the polymerase chain reaction test, so the PCR, but it's very limited in its availability. And then how to treat it, since Legionella is intracellular pathogen, you would treat it with antibiotics. And here's a recommended list of antibiotics. So Who is like at high risk to contract the disease? So actually less than 5% of people who are exposed to Legionnaires do not Legionella do not develop the disease. So it's actually not that contagious, but about 1 in 10 people who contract the disease do die. Some people who are in high risk are people who are 50 years old or older. Also, there's people. There's been studies that's been shown that more men than women actually contract the disease. It's about like a 3-to-1 ratio. People who smoke heavily, drink heavily, who have diabetes, chronic respiratory disease, chronic kidney disease, immunosuppression due to cancer or organ transplantation. So it's just people who have like weakened immune systems that are more likely to catch this disease. So how to prevent it? There's actually no vaccines for Legionnaires. So the key to prevent the disease is to minimize the growth and spread of Legionella. And the importance is maintaining clean water systems and implementing controls for Legionella growth. And so they do this by like presenting disinfectants. Like for example, in a swimming pool, they inhibit the growth of Legionella by using an adequate amount of chlorine. They would also control temperatures that's above 50 degrees Celsius for hot temperatures, and then lower than 25 degrees Celsius for cold water. And then they would also reduce stagnation of water, which is like not keeping the water still, by flushing unused taps. So where does this disease occur? So it basically can occur anywhere in the world when conditions are right. They often the cases of disease are not reported because it's often misdiagnosed. So there's an estimate that the true number of Legionnaires disease cases may be 1.8 to 2.7 times higher than what is actually reported. And so more cases are actually being reported. And so as you can tell on this graph, like nearly 10,000 cases of Legionnaires disease were accounted for in 2018 in the US. And they have a couple theories for this. They think like, oh, there's more awareness and testing for the disease, or like more people are more susceptible to this disease, or that there's an increased growth of Legionella in the environment now. And another fun pattern is that more illness is typically found in the summer and early fall, but it can also happen in the other seasons as well. Okay, so some outbreaks. So since the disease is not as reported as heavily, they believe that there is at least 20 outbreaks reported each year in the U.S. Outbreaks are commonly associated with buildings or structures that have complex water systems, like hotels or even cruise ships. There was a recent outbreak, one of the higher outbreaks actually, that in 2016 and 2017, 51 confirmed cases of Legionnaires disease were associated with traveling to Dubai. Another bigger outbreak was in 2015 when a cooling tower in Bronx, New York was associated with 138 cases of Legionnaires disease and killed about 12 people. And because of this, Bronx actually promoted for legislation to pass more control implements and maintenance of cooling towers and other water systems. And then a more recent outbreak was in May 2022. 5 people contracted the disease and were actually admitted to the hospital in Sydney Central Business District in Australia. And so the outbreaks are not too big, and it's very rare to have like bigger outbreaks. Here's my works cited. Thank you.